welcome to our GeoCast Summit. What I'm going to do is explain the format for this summit, how this is going to go for this afternoon, and then we'll get into the discussion. So the way the format's going to work today is in the GeoCast Summit, we'll do the first maybe 15, 20 minutes where I'll introduce you to the technology, the GeoMx Digital Spatial Profiler. It's a platform we just announced a full commercial launch at AACR about a month ago. And I'll give you an overview of what the platform looks like and how it works. And then at 1 o'clock, we will start to broadcast a session across North America on the West Coast. And so you'll see on the map there's multiple sites. There's folks on these different sites and folks online as well that will attend. So you'll have hundreds of attendees online that are listening into our broadcast. And we'll have two speakers, John McPherson, who's here, and Christina Curtis will be coming soon. And so their two talks, and I'll describe their talks in a little bit shortly, that'll get broadcasted live going outwards. And so the only other thing I'll point out is I'll do the introduction. It'll be much more casual for the first 20 minutes. We'll probably get a little more formal in the second hour because it'll be a tight timeline to broadcast the presentations. Is that as you have questions, there'll be two folks on the side with mics. Just wait for them to bring the mic to you because then the folks on the other satellites can also hear you in terms of what questions you're asking or answering. Okay? With that said, thank you again for coming to the session. It's really exciting for us. It's a very new format to share data. What I'm going to do is start with, I'm going to skip some of these. Okay. Start with giving you a basic overview of the platform and what it does. And then in terms of applications, John and Christina will talk about how they're using the platform and the capabilities in their particular studies. So the first thing I'll start with is, for a long time in NanoString, we've been looking at gene expression profiling in bulk assays. And what we've done is try to do these gene expression profiles really for translational research. How do we identify biomarkers and signatures that can select patients? And so this is the work that we did with Merck to identify an 18-gene signature that's predictive of a tumor being hot or cold, if you will. And so on the graph on the bottom, what you're looking at is when we look at these samples, with the signature that we've designed through bulk gene expression, we can segment patients by whether they have a tumor hot or tumor cold and if they respond to therapy. But what I wanted to point out was the blue arrow on the bottom, which shows you patients who are immune hot but are not responding to immunotherapy. And the question is, we have all these analytical tools, these bulk expression analysis, single cell analysis, what have you, but we're not able to explain the biology and understand why those patients are not responding. And so this is really the impetus for us to be able to design a spatial platform. And the idea is that can the spatial platform capture this heterogeneity in a way that bulk analysis cannot. And so when we initially thought about it, we look at IHC or ISH assays or gene expression profiling. And sometimes when you look at these patient samples by IHC or by bulk expression analysis, they may look very similar, yet they're responding very differently. And the idea is that if I have to go spatially and look at specific parts of the tissue, would I be able to unearth this heterogeneity that might explain where that difference is coming from? And so when we thought about that, we looked at these two worlds historically that we've lived in. One is the imaging world, which gives you this rich, colorful data set at a high level of resolution, but for a small number of targets. It may not be as quantitative as we want it to be, like expression profiling is. It may not be as precise. But then you've got this other world where in NanoString we've lived in for the last 10 years, which is really sort of high-plex expression profiling, which is reproducible, it's quantitative, but it really loses all that spatial information. And so the philosophy behind designing the GeoMx Digital Spatial Profiler is, can we bring both those capabilities under one roof so that we're doing imaging and profiling in a single assay? And so that's what makes that platform very unique. And so what we started with was that we also don't want to be able to compare data from two different chemistries and two different platforms. How do we harmonize this so that you can increase your signal to noise, if you will, and you're not focused on technical variabilities? And so the way we designed that, we started with a single barcode chemistry that then allows us to detect RNA or protein, so we want to be analyte agnostic. And then we designed that on two separate platforms that really talk to each other. So the nCounter platform is what we've developed 10 years ago that does expression profiling from bulk samples, so that's your standard expression profiling platform. We've connected that to the new digital spatial profiler that gives you the spatial information. So those two instruments together give you spatial and non-spatial or standard expression profiling data sets. And so the philosophy here is we take the reagents, whether it's a standard antibody that we use for IHC or probes to do ISH assays or standard gene expression profiling, we tag them with a photocleavable linker that's attached to a 6-temer DNA sequence. So you're seeing an antibody over there or an RNA probe that's tagged to a linker. We bathe that over a tissue, And now you have these profiling reagents that are with photocleavable barcodes that bathe the entire tissue. Then we identify regions of interest, we shine light on them, release those barcodes, and we can digitally count them. And the data output you get at the end is an image and a heat map. So you get both kinds of data sets. You get the full whole slide, single cell resolution tissue image under 4 colors, and then you get this ultra high plex, quantitative, reproducible profiling data set as well that's now spatially oriented. And so what's important to point out is because we do these 2 assays in one, we also have 2 sets of reagents. You have to have imaging reagents and profiling reagents. And the imaging reagents, the system has a 4-color scanner, so it's up to 4 antibodies or ISH probes, if you will, that'll have 4 fluorescent dyes. That's what you use to image the tissue and triangulate to the region of interest you care about. Then on the second, you've got these profiling reagents that have the photocleavable linker and the 6-temer sequence. There is no fluorescent molecules here, so it's not imageable, it's not visualizable. Cannot be visualized, right? And so you mix those reagents together, you bathe your tissue, and what allows you to do is then image and profile that particular tissue. So this is the actual workflow that we'll go through. In step 1, you bathe that cocktail with a combination of imaging and profiling reagents. It's up to 4 imaging reagents and really sort of an n-plex for the profiling. I'll talk about why that is. Then, using the instrument, you will image the entire tissue up to 4 colors. You will identify the regions of interest you care about, shine light, that's in step 3. You will shine light to only the regions you care about, release those barcodes. Then in step 4, you'll see a sipper that sits on top that picks up those barcodes and deposits them into a 96-well plate. Now, in that plate, every well has these pure 6-temer sequences, so there's no sample in this well, it's just the barcodes that are there. So you've separated the readout from the assay. And in that well, you've got barcode that encodes for not just target of interest, but also the space, where in the tissue it came from. And so that entire plate is indexed. Now what you can take is take those 6-temer sequences, hybridize to the probes, and count them on the nCounter platform. But they're also just 6-temer sequences, so you can actually sequence them on a sequencing platform as well to then reveal what the target is and where in space it came from. And so the barcoding chemistry that we use for the nCounter, if you're not familiar with it, there's 2 reagents we synthesize in Seattle. One's called the capture probe, one's called the reporter probe. The reporter probe is the molecule that we actually read out. And so it has 6 spots on it. Each spot can take one of 4 colors. So the combination is 4,096 barcodes. We've commercialized 800 of them. So on the nCounter platform today, we can count up to 800 different molecules. And those 2 ends there, they have 50 base pair sequences that overlap and bind to the target of interest. And that tripartite complex gives us the specificity of the molecule that we're going after. Now, this could be for a standard mRNA molecule that's in your sample of interest, but it could also be designed for the tags that are in the back of the antibody or on an RNA probe. And so what we do is take these sequences, the 6-temer sequence on the back of the antibody or on the ISH probe. We then hybridize that to 2 molecules, the capture and the reporter. We put that in the nCounter platform, and we count the molecules. So this is not looking at fluorescent intensity. We look at the sequence of colors. So if it's a blue, yellow, red, it could be p53. If I switch the blue and the yellow, it might be EGFR. And so the color sequence tells me what that particular target is. So when I count, I'm actually counting single molecules to figure out how many targets are in my particular sample. So this is one of our first experiments, probably did about 2 years ago, to really convince ourselves we're measuring what we're seeing. So we took a lymphoid tissue, stained it. In this case, it was Ki67 and CD3. And then what we said is I can visually see it. I can quantitate that immunofluorescence if I want to. But what we did was we took a barcode, and for these rare experiments, we tagged it on one side with the oligosequence and the other side with the fluorescence. So with the same antibody in this experiment, I can visualize it, but then I can shine light right to that spot, release those barcodes, and actually digitally count them. And that's what you're seeing in that graph, is actual digital counts, molecule by molecule, what's in that region of interest. But when I do these profiling molecules, I'm not looking at 1 or 2 molecules, because then I'm not limited by how many channels I have. I'm just counting them. So what I can actually do is tag a lot of different molecules, and in any region of interest, I can create these massive heat maps. So expression profiles that deeply characterize a particular region in the tissue. And so that's what the DSP really does. It spatially profiles in a tissue that's guided by the imaging reagents. But what makes this really interesting is that the entire performance of that platform really depends on the control of your light source, because your light source is essentially a scalpel that identifies the regions you can assay. And so the tissue is very irregular, so you have to have a system that has that level of resolution to carve out only the parts of the tissue that you care about. And the way we do that is we use something called a digital micromirror device. The one we have is smaller than a postage stamp. It has about a million mirrors in them. Each mirror is about a micron by micron. And so each mirror can be individually controlled to shine light. So it's almost like a flashlight. Each mirror can shine light in a particular part of the tissue. So what I can do is I can take a tissue microarray core like I've done here, stain that for tumor and tumor microenvironment, and use that staining pattern and a threshold to create a binary image of ones and zeros. I take that image, I can pass that to the DMD, and the DMD will conform to the irregular organic shape of the tissue and be able to release the marker. So for example here, I can create a ones and zeros of mask 1 or the inverse, and I can then do in 2 moves. In the first one, I will release all the markers from all the white region. In the second, I can do it from the black region. And now I can get an entire population of a tumor microenvironment, tumor and the tumor microenvironment separated out of the tissue, and I've got 2 pure profiling data sets. I can also program the digital micromirror if I wanted to walk these islands. So I can start with a small white island on the bottom and segment by segment tease that out so I get even higher level of heterogeneity or resolution from that data set. And so that's really the promise of the digital micromirror. So you can really convolve to any shape size you want, so you can tease out these organic shapes out of a tissue sample. And so what we then did was we took the digital micromirrors and we trained them in very specific use cases that actually matched the biological questions you want to ask. And so starting on my left, the easiest one to start with is always the geometric. It's pretty simple. You'd find pretty homogeneous regions around the tissue. You draw circles and squares and profile that. The light shines and releases those markers. You may find in certain tissues there's a lot more interdigitation of the cells, and it's hard to tease them out with circles and squares because you're going to get mixed populations. And so when you have a fluorescent image, just like I showed you, you can do image thresholds, create the binary mask, and tease those things out into pure populations. Sometimes what you may care about is not that region of interest, but some rare cell population that lives in that region of interest that you want to profile. It could be Tregs and macrophages or what have you. And as long as you have markers that identify that rare cell type, you would stain, pick out those cell types, and the DMD simultaneously, almost like a strobe light, would hit all these different cells and release those markers, and you'll pick them up and count them. So you can, the first pass, you can do macrophages, and the second pass, you can do Tregs, and so on. The fourth one is the contour mapping. And so the contour mapping really is about if you have a particular structure in a tissue and you're looking to see how that cell structure is impacting expression levels in a distance-oriented or proximity-oriented way. So for example, let's say you have a tumor and you have an invasive margin. Well, you can create all these contour maps that go out 50, 100, 75 microns out, and you can start to look at the immune expression profile as you move away from the tumor. So it allows you to characterize some sort of an expression pattern that's dependent on some cell structure you may have. And the last one is a gridded. That's like, if you have a tissue sample, you really don't know what you might be looking for. It's a discovery experiment. You can unbiasedly grid it and then profile all those samples. So typically what we would do is, here's an example of the data sets that come out of a study like this. The first one's a lymphoid, the second one is a colorectal sample. And here we've done something very simple. Put geometric shapes where we think there's uniform areas of the tissue. We profile it. These are protein profiles that we're doing. And what you start to see is a tremendous amount of heterogeneity across region by region, just because there's a different composition of cells, different biology that you would lose if you're doing bulk expression analysis. But typically what we would do, and in the next slide I'll show you what typical experiments look like if we want to maximize the amount of data you get and maximize your chance of finding a biomarker. And so in this case, we did a colorectal cancer sample where we had MSI and MSS status. What we also did here was used a bulk gene expression signature, the one I showed you before, the tumor inflammation signature, to figure out which of these tumors are hot and cold. So we have bulk gene expression data sets, we have MSI, MSS status as well. Then we took these samples and we did both RNA and protein analysis. And so we did about 40 proteins, 84 RNA. These are all in FFP samples. We do them on sequential sections. And what we did here for the region of interest, that we would go into these particular tissue samples, and this particular sample is highly interdigitated, so we couldn't use geometric the way we wanted to. And so we decided to do segmentation. And the software automatically segments this for you based on the threshold patterns. And so what we can now do is take sequential sections. On the first one, I select my regions of interest. I superimpose that to the second slide as well. So one slide's for protein, one slide's for RNA. I do the segmentation on the first one and the second one. Now I've got mirror images of RNA and protein profiles from those particular tissue sets. And so then you start to generate these heat maps on the RNA side and the protein side. I can start to cluster them by their TIS signature for immune hot or cold. I can segment them by MSI, MSS status as well. And so that's how we start to bring in our bulk analysis and spatial analysis together into one single analysis tool. So I also pointed out, in the way the system works is that the GeoMx platform, it's really an imaging platform. It creates a 4-color image, and the output is a plate of barcodes. Those barcodes can go into an nCounter platform that has barcoding capacity up to 800, that we can measure up to that many targets. Or you can take those barcodes into a MiSeq, NextSeq, or a NovaSeq if you want, and sequence those as well. And what's nice about that is it opens up your barcoding capacity. You're not limited to 800 anymore. And so when we have these two types of readout capabilities, we can start to think differently about our content design. Because it may be that you may want a 30-plex proteins, and you may want a 50-plex proteins or 100. And we want to think about flexibility in how we design the content and the applications. So one of the unique designs in the content is that on the protein side, we find biological domains, and we identify core sets of targets that most people may want to assay. So in this case, for example, in immuno-oncology, we've designed a core that's about a 20-plex target that really looks at cell typing, if you will. And then what we do is create these 10-plex modules that can be spiked in. They're all validated together. And so what you can do is run an assay of the 20-plex by itself, or you can spike in 10-plex at a time. And you can spike in up to 6 of these panels. That gets you up to 80-plex protein targets. And there's an additional 10-plex that's left open. If the targets are not in our panel, you can customize that 10-plex and then spike that in. So you can get up to about 90-plex on the protein side. But you can build it as you see fit. And so that's how that design is set up. On the RNA side, we've done it in 2 flavors. One, with RNA, we've designed sort of 80-plex assays to match the proteins that can be read on nCounter. But when we get to NGS land, we can think completely differently about the barcoding capacity. And so there, we're starting to build our first set of NGS-driven RNA panels. And the idea is we want to get to the transcriptome as quickly as possible, but we're going to start doing it in chunks of 500. So we're trying to identify biological areas. We're not limited to 500. It's arbitrary. And so we've identified 500 sets of genes that describe the tumor, a set of 500 genes that describe the immune compartment. And what you can start to do is you can profile 500 genes at a time, or you can collapse them all together to 1,000 or 5,000 genes. All these panels can be put together, integrated, and you can build them as you go as a Lego set, and then be able to sequence that spatially profile. And today, John will talk about, he was one of the early testers of the panel, so he'll share some of the data that he got from the 1,500-plex assay that we ran early this year. So the last part of it, I'll focus on the software. So on the software, this is a very unique software that we had to design that's unlike any other because the data set that comes out of it is different. Because we have spatial data that's being generated on one side, the 4-color image, and then we have this expression profiling data set that actually comes from specific regions of the tissue, we have to somehow figure out a way to connect those things intimately so that that link is never broken. Because imagine the profiling data actually travels outside of this instrument, goes into another sequencing platform, has to come back in. And that's what the software is really designed to do. There's 2 parts of the software. One's called the control center. It's really designed for a technician to be able to come on and push button. A histotech could be able to run this platform. It's very visual and video driven to go step by step to run the assay. But once the data is generated, the data analysis tool is fairly complex. This is really designed for a scientist to interact with that data set. So you'll see the look and feel of the 2 data sets are different. But you'll also see the GeoMx platform generates a 4-color data set that feeds in the data analysis. The data analysis then accepts the profiling data from a completely different platform, but it never forgets the connection between the image and the profiling data set. And that's what the software does. It creates that data fidelity so that you never lose that connection. And the reason that becomes powerful is this is what the face of the software looks like when you interact with it, is the interactivity of the data set. So for example, you have images on one side, you've got the heat profiling on the other side. And let's say you're partial to the profiling side of the data, you're analyzing the profiling side of the data, you identify a particular heat map that you're interested in. You can click any bar on that heat map, and the software will pull up the image and highlight the region of interest that data came from. And if you're partial to the image side of it, more familiar with it, you can identify a region of interest and click on it. It will pull up the heat map or the expression profile from that particular region of interest. And you can go back and forth and interact with that data set all day long. And that's really hard to do if these two data sets live somewhere else. You have to have an Excel sheet that sort of tracks them together. And this does it automatically for you. And that's the unique feature about the software. And so because it has that unique capability, we have to then build an ecosystem to bring all your external data in here so you can overlay that, so you can look at that data in aggregate. And so the way the software is designed, right now the software works on-prem on your instrument. And then early next year, we'll launch the cloud capability. And what this allows you to do is a couple of things. This is designed to be very interactive, because you can imagine if there, let's say, is an instrument here, and there's a technician running it, they're selecting regions of interest. It may be that they want someone from Boston to select the regions of interest. And somebody in Boston can log on to a web page, access the software on the instrument, and they can select region of interest and hit go. Right? And so, or a technician here could pick the regions of interest, send that for approval to any scientists around the world. They can review it and approve it before the experiment runs. And that's just in the experiment design, selection, running of the instrument. But once the data is generated, the data is also interactive. You can have multiple users logging onto a web page, analyze the data, and save their data differently, and start to compare data sets. So it does not mean that you have to have a desktop. You have to be in front of the machine to do it. You can do the analysis anywhere. You can share data anywhere, because the data sets are just so different. And allows you to import data sets and scripts as well to do custom analysis. It's very flexible in that sense, just because of the richness of the data that's being generated. So what's unique about the platform is a couple of things. We've tried to be analyte agnostic. We don't think about this as an RNA or a protein platform, because biomarkers can come from anywhere. We've really tried to push the limits on the plex, because we don't want that to be limiting either. And that's why opening up to NGS readout really allows us to go as many targets as possible. Today on the protein side, we pushed up to the 90 plex, because for proteins, that is like the transcriptome. On the RNA side, there's not a lot of constraints as we see on the protein side. So there, we are pushing it towards a transcriptome. So in the near future, we can start to spatially profile the transcriptome. Because we're digitally counting these as single molecules, these are quantitative platforms. The resolution gets you down to 10 microns for the, on the protein assay, about 100 microns today for the RNA assay. And it's non-destructive. The entire thing uses light to profile the sample. We don't laser ablate it. You don't cut the samples. The sample's actually reusable. You can strip the antibodies, reprobe them if you want. The same sample, once it's gone through a DSP experiment, you can do an H&E stain and visualize it. The same sample after the experiment, you can scrape the tissue and do DNA sequencing and do genotyping if you wanted to. And the last thing we focused on is the throughput. The throughput's important to us is that if you're looking at biomarker studies and we want to power these studies, you have to be able to push through tens and hundreds of samples pretty quickly. And if you have a really high plex imaging platform, it would slow down that throughput. And for us, in a single day, we can do about 10 to 20 slides a day with this level of high plex capability. The last two things I'll quickly point out is that at AACR, we announced a series of partnerships that help us develop the platform to the next level from where we launched it. There are three partnerships, Leica, ACD, and Abcam. And these partnerships are really important because they do things that we don't necessarily do. So for example, Leica can allow it will develop an automated sample prep, so these slides can go through for a fully automated sample prep, and that'll happen by the end of the year. So we can push button, run all these protocols on a Leica autostainer. With ACD, they have some fantastic RNA imaging reagents, if you've heard of them, the RNASeq reagents. Those are very complementary. RNASeq and our RNA chemistry are the only ones that work on FFPE. So we'll integrate these reagents so the RNASeq reagents can allow us to visualize the tissue, and then we will then do high-plex profiling with our reagents, and that'll be one integrated solution that'll be available. And the last one, what I'm really excited about, is also Abcam. It's really hard to work with antibodies, and so working with companies like Abcam, who have over 6,000 antibodies that are validated for FFPE, allows them to create a large menu of antibodies very quickly that you can pick and choose and run your DSP assay. And so our goal really is to be able to integrate these two data sets. It's not about doing standard expression profiling and looking at spatial information. We think these things generate different kinds of information. We're already seeing that today. And so the idea is, how do you create an ecosystem? You can bring your standard expression profiling data sets, your single cell data sets, and how do you integrate that with your spatial profiling sets to increase the chance of finding a biomarker? And so that's really the goal of the platform. In the 30 seconds I have, I'll just point out, if you're interested, there is a manuscript in bioRxiv that fully describes the platform, how the RNA chemistry is designed, how the protein chemistry is designed, how it's validated. There's application data sets in there. So if you're thinking about writing grants and so on, or really want to understand the technology, the manuscript goes in enormous depth, explaining every aspect of the technology, including the validation of how the DMDs work and their resolution and so on. And so if you can't access it, let me know. We'll get you a copy of that manuscript as well. So with that, I have 10 seconds left. I'm going to pause. We're going to probably take a quick 2-minute break, and then we're going to get started with John and Christina on talking about their studies and their experience. So those will be actual case studies about using the capability. And that will then obviously broadcast. They'll also be available recorded afterwards if you want a copy of it as well. So we'll take a 1 to 2 minute break, and we'll come back here and we'll get the second session started. Thank you.